my name is Ali Shaladi. I'm an investigator at the Starr's Institute, and my laboratory studies the mechanism of chromosomal translocation and cancer pathogenesis. Why is it that a single point mutation in a general transcription factor results in pathogenesis of a specific type of cancer? Work from my lab and other laboratories have suggested that enhancers could play a central role in this process. So enhancers are DNA elements that regulate tissue-specific expression pattern of a given gene. So for years, scientists across the globe have been looking at the expression pattern of a gene and the mutations within that gene that correlate with cancer. But now, since we are able to look at the entire genome because of the advent of new technology of genome sequencing, what we have learned is that these intergenic sequences that are not within the protein coding gene, which they only constitute about 2% of our genome, these 98% are highly mutated in different forms of cancer, and some of these known enhancers are also mutated, and their mutation correlates with different forms of cancer. This work is very important because coincidentally, the work that has been done in sequencing the cancer genome has shown that factors that are involved in monomethylation of enhancers, as a compass family member, the MLL34 that my lab has identified, are hit in large number of cancers, including from bladder to brain to breast to kidney to colon. Large number of solid tumors have mutations within the MLL34 and the compass family. Given the fact that we have shown that MLL34 are enhancer monomethylases, they're functioning enhancer promoter communication, and now they're hit in large differ of cancer, we're proposing that perhaps point mutation of these MLL34 could bring about tissue-specific cancer. So my colleague Hans-Martin Herz and Dae-King Hu and I have written a perspective titled Enhancer Malfunction in Cancer, which will be published in the March 20th issue of Molecular Cell. I'll also be presenting this idea of the enhancer malfunction and its role in cancer in the upcoming American Association for Cancer Research, AACR, in a Meet the Expert session where I propose this idea, and then we'll talk among colleagues who has found mutations of MLL34 in their type of cancer that it studies, and how our reagents and our tools and knowledge could be helpful for their research, and what they have found within the cancer studies that they can have done could be helpful for the research that we are doing in the lab. So this has been the most exciting time in my scientific career during the past 20 years. And our lab is working very hard and very focused on this model of enhancer malfunction in cancer. If the model turns out to be true, we would be able to inhibit this MLL34 compass family in many of these cancers that are caused by MLL34 mutation, and perhaps develop a targeted therapeutics.